Hi guys, and welcome to our module on Embryology Week 1. These are the various events that occur in the week 1 of embryology. That is from fertilization till implantation. Uh, Fertilization is the first step of the week 1 in terms of conception. That involves the union between a sperm cell and an oocyte to give rise to a zygote. And then this zygote will go ahead to undergo various processes involving cleavage, blastogenesis, and finally implantation. Implantation tends to begin at the end of week 1 and continue on to week 2. Starting with the very first stage or process involved in week 1, fertilization. Fertilization is a process of union between the sperm cell and the oocyte to give rise to a zygote. Fertilization tends to occur particularly within the uterine tube, or we call the fallopian tubes. The specific sites where we tend to have fertilization occurring at would be the ampulla of the uterine tube. That's the largest or the most dilated part of the uterine tube. There are three major phases of fertilization. Phase one involves the sperm penetration of the corona radiata of the oocyte. Phase two involves the sperm binding and the penetration of the zona pellucida of the oocyte. And then finally, phase three involves the fusion of the sperm cells together with the oocyte cell membrane. And at the end of the day, we have a mixture of the nucleus found within the sperm cell with the nucleus found within the oocyte, and fertilization becomes complete at that point. Talk about each of these different phases in much more detail in the next couple of slides. Starting with the phase one of fertilization, phase one entails the penetration of the sperm cells through the corona radiata of the oocyte. And what happens in this particular phase is that enzymes present within the sperm or within semen and the uterine tube mucosa are going to be useful in this penetration occurring. They help in breaking down the barrier that is the corona radiata of the oocyte. And in doing so, they allow for the sperm cells to penetrate through and come in contact with the next layer of the oocyte known as the zona pellucida. Phase two of fertilization involves the interaction between the stem cell and the second layer of oocyte, which is the zona pellucida. This interaction is much more of a direct contact than it is for a penetration. So the sperm cells tend to bind to the zona pellucida through the use of various protein receptors. So on the sperm cell, we have what they call the um, glycosyl transferase receptors or glycosyl transferase protein. Whereas on the zona pellucida, we have a particular set of receptors called the ZP3 receptors, also called zona pellucida 3 receptors. These two major structures, the one on the sperm cell and the one on the zona pellucida, would interact with one another. So the glycosyl transferase protein on the sperm cell will interact with the ZP3 receptors on the zona pellucida. And once that interaction occurs, once that binding occurs, this triggers a reaction we call the acrosome reaction, and that leads to the release of enzymes we call acrosomal enzymes. It is these acrosomal enzymes that will now further lead to the penetration of the sperm cells from the zona pellucida into the cytoplasm of the cell. And that is very important to remember that the acrosomal enzymes occur when there's the binding between the sperm cell and the zona pellucida, and that binding leads to triggers that reaction we call the acrosome reaction. The acrosome reaction is not the only reaction that causes that. After the sperm cell has bounded to the zona pellucida and the acrosome reaction has occurred, that acrosomal enzymes are released, and these acrosomal enzymes are usually called acrosine. These enzymes would help in the further penetration of the zona pellucida. And how this occurs is that the acrosomal enzymes themselves trigger a secondary reaction. This reaction is called a cortical reaction. And what it is important to note about this reaction is that once a single sperm cell binds and triggers the acrosomal reaction, releases the acrosine, and starts to penetrate through the zona pellucida, the protein that is being produced or being released that triggers the secondary reaction, the cortical reaction, would allow for, or rather disallow for, other sperm cells from binding on the that single oocyte. So if we have a single oocyte and a single sperm cell, and one sperm cell gets to the oocyte first, binds, triggers the acrosome reaction, which triggers the cortical reaction, This will prevent other sperm cells from binding further. And what happens practically is that the cortical reaction will render the solar pellucida and the oocyte of the membrane of the oocyte to be impermeable to other sperm cells. Therefore, we have a single sperm cell to a single oocyte occurring in fertilization at the end of the day. Final phase of fertilization is phase 3 involves the fusion of the sperm and the oocyte cell membrane, and for the sperm cells to penetrate through the oocyte and get this phase, needs to go through the cellular barrier to penetrate through the coronal radiata in phase 1, then penetrates through the zona pellucida in phase 2, and finally, the membrane of the sperm cell comes in contact with the membrane of the oocyte, and this undergo fusion. So this tends to lead to the entire cytoplasm of the sperm cell mixing with the entire cytoplasm of the oocyte, and then we have just a single cell at the end of the day. So once the cell membrane of the sperm and the oocyte fuses together, five major things follow. The first is that the sperm cell loses its neck and its tail. The neck of the sperm cell contains mitochondria, and this is lost during fusion. The tail contains microtubules that helps in the propelling or movement of the sperm cell, and this is also going to be lost after fusion occurs. Second thing that happens is that the nucleus of the sperm cell becomes what we call a male pronucleus. For the nucleus of the oocyte, before it can become a female pronucleus, the third thing that must be that nuclei must undergo almost complete meiosis. So remember that the secondary oocyte is stopped at the metaphase stage of meiosis two, and it's at fertilization that after the fusion of the sperm and oocyte occurs that this meiosis two becomes complete and the oocyte undergoes full maturation into an ovum. Then from there, the nucleus of the ovum will become the female pronucleus, and then the male and female pronuclei would fuse together to form a full blown zygote. Remember that as we talked about with the sperm cell, the amount of chromosomes present within the nucleus is about 23, so the haploid in number. For the ovum at maturation, the amount is also 23, that is haploid in number. And once these two fuses together at the end of fusion of phase 3, the zygote that is formed will contain a total of 46 chromosomes in its new nucleus, which is a fusion of the male and the female pronuclear. And that's one of the reasons why we say that 23 chromosomes comes from the father, which is from the sperm cell of the father. 23 chromosomes come from the mother, which is the oocyte of the mother, to give us a total of 46 chromosomes. This picture explains in a nutshell all we discussed about fertilization. Remember that it starts with phase 1, not shown in this image, but phase 1 entails the interaction between the sperm cell and the corona radiata. Then that is followed by phase 2, in which the sperm cell, after penetrating through the corona radiata, comes in contact with the zona pellucida, as shown in the image here. That contact will lead to the triggering of the acrosome reaction. The acrosome reaction entails the release of various hydrolytic enzymes, what we call acrosine. These enzymes further helps in breaking down the solar pellucida and allowing for the um, entry or penetration of the sperm cell. As the sperm cell penetrates through the zona pellucida to enter towards the perivitelline space, a second reaction is triggered, and that reaction is called the cortical reaction. You can see cortical granules being released, and this will prevent other sperm cells from permeating or penetrating through the zona pellucida. So it renders the oocyte to be impermeable to other sperm cells, aside the initial one that was that came in contact with it at phase 2. And after that, we have phase 3, whereby the plasma membrane of the sperm cell and the egg would fuse together. This leads to a number of things. The sperm cell loses its neck and its tail. So tail and the neck is lost. So the mitochondrion in the neck is lost. The microtubules in the tail is also lost. The cytoplasm of the sperm cell fuses with the cytoplasm of the oocyte, and thereby releases its pronucleus, or rather releases its nuclei into the cytoplasm of the oocyte. At this stage, the nucleus of the sperm cell begins to be called a pronucleus. The other thing that happens in the oocyte is that we have completion of meiosis 2 from the secondary oocyte, and from there, that nucleus of the oocyte starts to be called
the female nucleus, and then finally fusion of the male and female pronucleus to give rise to a 46 chromosome or diploid zygote, which we'll talk about next in terms of what happens. The next step of processes that occur after fertilization has been completed is the stage we call cell cleavage. The cell cleavage is simply a series of mitotic divisions of the zygote that occurs in succession. So zygote cytoplasm successively partitions or cleaves to form a number of cells. So we start with a single zygote, that is a single cell shown here. That cell undergoes mitotic division to give rise to um, two cell stage zygote. Then this undergoes further division to give rise to a four cell stage zygote, and then further division occurs to forming an eight cell stage zygote. Each of these cells together are known as blastomeres. So if you look at this, we have to have a single cell. After further mitotic division, you have a clumps of cells that are clustered together. Each of these cells are known as blastomere. And at the end of the day, we have the zygotes that are multicellular in form. So blastomeres are considered as totipotential cells, or as that is from the fourth to about eight cell stage. But once we get to the 16th cell stage or blastomere, we start to refer to the zygote as a morula. And this is a special term that we use in referring to this from. So any of the zygote that is 16 cell stage and above is referred to as a morula. And we'll talk about the various things that the morula tends to undergo in the next couple of slides. At day three, the morula tends to mature, undergoing a particular process we call compaction. So, compaction, think about compaction as each of those different cells, those blastomeres, coming into tight contact with one another and being very well held together and um, sealed off from the external environment. This happens because of the formation of what we call tight junctions. So, tight junctions, as discussed in a previous module, are specific intracellular junctions that tend to connect tightly cells that are well aligned, that are found close to one another. So in the case of the morula, the 16 to 32 cell stages of the zygote, all those cells that you see clustered together are eventually compacted, and this is done by the formation of those tight junctions, and then they are sealed off. So this occurs within the third day of um, conception, and followed by which we start to see further development of the morula from a morula to what we call a blastocyst. And we'll talk about that in the next slide. This image just depicts for us the various events occurring from day one all the way to day three and beyond. Starting with day one, we have fertilization occurring. Remember that fertilization occurs largely at the ampulla of the uterine tube, as shown here. That's the largest that largest aspect, widest aspect of the fallopian tube. After fertilization occurs, a zygote is formed. The zygote undergoes further mitotic division to give rise to a two-cell stage. This forms eventually a four-cell stage through further mitotic division. Then from there, we have an eight-cell stage zygote. From this, the eight-cell stage further developed into a 16-cell stage zygote. In this case, we start to call it a morula, no longer just a simple zygote. And the morula further undergoes development through the process of compaction and automatically would become a blastocyst, which we'll talk about next. And that will be the stage at which the zygote tends to be implanted. By default, the blastomere or the morula tends to undergo the process called blastogenesis. So blastogenesis just involves the formation of a fluid-filled cavity within the morula. Remember that before this particular process occurs, the morula is just clumps of compacted cells found within the zygote, and once we start to see fluid present or a space containing fluid present within the morula, all the cells displaced to one side, that morula ceases from being called a morula. We start to call that a blastocyst. So blastocyst has cells and has a cavity. The name of the cavity we see in the blastocyst is called the blastocele or the blastocyst cavity. I'll show you a picture of how that looks like very soon. The blastocyst contains a number of things. It contains the inner cell mass, an outer cell mass, contains um, the fluid-filled space known as the blastocele. The inner cell mass of the blastocyst is usually also called the embryoblast. It is what would eventually give rise to the fetus of the embryo. The outer cell mass of the blastocyst tends to be called the trophoblast, and it tends to give rise to the part of the fetus we know as the placenta. We'll talk about that very soon. This picture captures for us all the various events from day 1 to day 4. From day 1, we have a zygote being formed, a single cell structure, and this undergoes cleavage to form a 2 cell stage, the 4 cell stages, and 8 cell stages of blastomeres. Blastomeres will undergo further division to form the 16 cell stage morula. At this stage, the morula would undergo compaction. Compaction in this case would be the tightening up of the blastomeres and the sealing off from the external environment. After the morula is being formed, around the 4, the morula starts to undergo the process we call blastogenesis. And what you notice is that from these clumped clusters of cells that you can see in the morula, we have a large space being formed. That large space is called the blastocele or the blastocyst cavity. And you also notice that the cells are also directly displaced and arranged in a very unique manner. We have some cells that are clustered together at the edge here. These cells are known as the inner cell mass or the embryoblast. These are the cells that will give rise to the embryo. And we have some cells at the edge of the edge of the zygote. These cells are known as the outer cell mass. These are the cells that will give rise to the trophoblast or the placenta in the fetal organism. So this particular cell form of the zygote at this stage is known as the blastocyst because of the presence of that fluid-filled cavity. The final process that occurs at week 1 is the process of implantation. Now, implantation begins at week 1 but is completed at week 2. So remember that at day 4 we have the morula that is undergoing blastogenesis to form a blastocyst. So at the end of day 1 and day 4 we have a blastocyst being formed. This blastocyst still contains a zona pellucida, and around day 4 to 5 we start to see the zona pellucida degenerating. This degeneration of zonal pellucida is very important for implantation to occur, and as the zonal pellucida degenerates, it's also important to know that the blastocyst has moved or is moving from the site of fertilization at the ampulla towards the uterine tube. Usually at day 5, the um, events that occur at day 5 is that the blastocyst would have moved into the uterine cavity and the zonal pellucida tends to start to degenerate at that point in time. Once the blastocyst is found within the uterine cavity, it tends to implant within a particular aspect of the wall of the uterus. It typically inserts at the posterior superior wall of the uterus, and this occurs around day 7. So implantation tends to begin at day 7, and implantation occurs in the functional layer of the endometrium, usually during the progestational or secretory phase of the menstrual cycle. This is a picture showing for us all the things we talked about from the point of fertilization all the way down to implantation. And you notice that the implantation is practically the embedment of the blastocyst into the uterine wall. As you can see, this is the uterine wall here in pink. This is the blastocyst here in purple and green. And you can see that they are interacting using various receptors. As shown in the image, the blastocyst tend to implant into one, uterine wall, and from there further development will occur at week 2 to week 3 and all the way till the 9 months. One important clinical significance to take home from week 1 is a process or pathology known as ectopic pregnancy. Now, ectopic pregnancy is an abnormal implantation of the blastocyst. Normally or typically, blastocysts tend to implant at the uterine wall, but if the implantation occurs anywhere else aside from the uterine wall, we know this as ectopic pregnancy. So this could either be at the uterine tubes themselves, so if the blastocysts fail to move from the ampulla towards the uterine cavity, it could implant in the uterine tube, and that's the commonest site of implantation. It could also implant all the way down to the cervix. At the cervix, it could be implantation occurring there. The implantation could also occur outside of the entire female internal genitalia. So imagine the if fertilization is occurring outside of the ampulla, it's occurring at the infundibulum or just at the pelvic cavity without entering into the fallopian tube. That zygote could or that blastocyst could implant outside of the uterus or outside or within the abdomen, and as a result, that could lead to an abdominal pregnancy. And that's very common. That's not really common rather, but that is very likely to occur. It's not an impossibility. So this is just one very important clinical significance. It's usually a very terrible thing to see a woman with ectopic pregnancy, especially someone that has been trying to give birth and having the child um, implanting at the at an unusual site. Usually, it's something that leads to urgent surgeries being done so that we can remove that blastocyst, preventing the woman from going into serious life-threatening complications. That concludes our module on embryology week one.